Welcome back to Breakdown of Science. I'm Jan.
I'm Maruf.
And I'm Michelle. So today we're continuing the part 2 of our discussion on gene editing and modification, also by covering some applications and case studies, and definitely going a bit deeper into how it works, and also touching on the ethical issues and the discussion surrounding this topic.
Yep, but first a quick recap of part 1.
Yeah, so the history, gene modification started with selective breeding, but real DNA manipulation began in the 1970s.
Yep, and then in 1953, DNA's double helix structure was described. And by 1972, Paul Berg created the first recombinant DNA, joining genes from different sources.
Yeah, and in 1975, Asilomar Conference set the safety rules for it.
Yep, and in 1982, the first biotech drug, insulin, made from bacteria, was approved.
And in 1990, the first human gene therapy was performed, which treated the immune deficiency, for example. Yeah. Now, in terms of the definitions, gene modification, it's basically inserting or deleting big DNA parts, sometimes also from other species.
And gene editing is making precise changes, like fixing a single gene.
Yeah, and knockout is basically turning a gene off, and knock-in is the other way around, so inserting a working gene.
Yep.
Yeah.
And then somatic means body cell, so not inherited. And then germline repeat means reproductive cells, inherited, and in this case, when it comes to gene modifications, it's very controversial.
Yeah.
Now, before CRISPR, we had the ZFNs and the TALENs, which were molecular scissors for cutting DNA.
Yeah. ZFNs, they're just zinc fingers that are used to find sequences, but they're definitely very hard to design.
Yep. And while TALENs were more flexible, flexible and used protein bricks, so to say, to match DNA.
Yeah, but both were definitely expensive and also very time-consuming to customize. So yeah, there's that.
And now CRISPR comes, and it was discovered through the bacteria's immune system. And in 2012, Dudan and Carpentier showed how it could cut any DNA using the Cas9 and guide RNAs.
Yeah, the key is that the single guide RNA, also known as sgRNA, tells Cas9 exactly where to cut.
Yep. And in 2013, CRISPR worked in human and mouse cells, which was a huge breakthrough.
And CRISPR is cheap, fast, and precise, and that's why it changed everything. Yeah.
Now, going into today's episode, so now that we understand how gene editing works, let's look at what it's actually doing out in the world. Applications range from treating genetic diseases to redesigning crops and even protecting entire ecosystems. Let's first begin with medicine. One of the most promising uses for gene editing is treating inherited blood disorders like sickle cell disease or beta thalassemia. Sickle cell causes red blood cells to become rigid and crescent-shaped, blocking blood vessels and causing severe pain, while beta thalassemia, on the other hand, results in not enough hemoglobin being made, which leads to chronic anemia.
Yeah, and in both cases, the problem lies in the mutations to the genes that make the hemoglobin. So one solution is to extract a patient's own hematopoietic stem cells, correct or disable the faulty genes using the CRISPR, and then we introduce those edited cells back into the patient. And this is called the ex vivo editing.
Yeah, one of the biggest breakthrough here is hexagamglycine autosomal cell, also known as Casgevy. It's the first CRISPR-based therapy approved by the FDA, and early results are amazing. Many patients treated for sickle cell or beta thalassemia no longer experience the symptom that once dominated their lives.
Yep. And then there's also in vivo editing, where CRISPR components are delivered inside the body to target tissues directly. For example, researchers are using AAV vectors to treat genetic forms of blindness by delivering CRISPR to the retinal cells directly.
Yeah, and when it comes to the liver, scientists are turning to lipid nanoparticles. These are just basically tiny fat-based capsules that can carry CRISPR tools safely into the cells. And one application is in reducing dangerous cholesterol levels by knocking out genes like PCSK9. Yeah.
Yeah, editing the liver is a key frontier, not just because it's metabolically active, but because many genetic conditions affect how the liver processes fats, sugars, and also proteins, delivering edits that there can have systematic effects.
Yep, and another exciting area is cancer immunotherapy. Here, CRISPR is used to reprogram T cells, which is a type of white blood cell, so they can better attack tumors.
Yeah, and this can mean that knocking out immune checkpoint genes like PD-1, for example, which not so PD-1 acts like brakes on the immune system, but are then hijacked by the tumors. So, or scientists might insert CARs, that stands for chimeric antigen receptors. They act like specialized sensors guiding the T cells straight to the cancer. Yeah.
Yeah. So what CRISPR does is CRISPR makes this process faster and more precise. Instead of inserting whole new genes randomly, it can target safe zones in the genome, which reduces the risk of complications like unwanted mutations, for example.
Yeah, and early trials using CRISPR-edited T cells, especially for blood cancer like leukemia, have shown promising results in both safety and efficiency.
CRISPR is also showing potential in fighting infections. For example, for instance, editing the CCR5 gene can make immune cells resistant to HIV, so similar to what was seen in the Berlin patient who was functionally cured of HIV after a bone marrow transplant.
Yeah, and researchers are even exploring ways to use CRISPR to target viral genomes directly, a sort of genetic antiviral approach. It's early stage, but it could revolutionize how we treat diseases like hepatitis B or even latent viruses like herpes.
Yep. And meanwhile, CRISPR is already being used as a diagnostics tool. Platforms like Sherlock and Detector, which uses CRISPR protein to detect viruses or bacteria by recognizing specific sequences in their DNA or RNA.
Yeah, and these tools were developed rapidly during the COVID-19 pandemic, so offering the fast and sensitive testing, so almost like a molecular radar system that can flag even trace amounts of infection.
Yeah. So we've talked about how the application is currently on humans, but we can also shift towards agriculture. So CRISPR is making crops and livestock more resilient, nutritious, and sustainable.
Yeah, in crops, the technology allows for precise non-transgenic edits, meaning no foreign DNA is inserted. So, for example, researchers have created a version of waxy corn by deactivating a gene that changes the structure of starch. So, great for food processing.
Yeah. Other examples include making tomatoes more resistant to fungal infections or engineering rice in a way that uses nitrogen more efficiently, reducing the need for fertilizers.
Yep, and that's a big deal for the environment. With less fertilizer runoff, that means it's less pollution and fewer algae blooms in waterways.
Yeah, and CRISPR is also helping to adapt crops to climate change. So by making them more heat or drought resistant, scientists can tweak regulatory genes that control how plants respond to the stress. Yeah.
Yeah. And when it comes to livestock, CRISPR has been used to create pigs resistant to RRS virus, cattle that are born hornless, and chickens with enhanced resistance to avian flu. So all without adding genes from other species.
And there's also interest in improving animal welfare. For example, editing genes to make fish grow faster or to prevent diseases means fewer antibiotics and less suffering in industrial farming.
And let's not forget gene drives, so CRISPR powered systems that bias inheritance. So instead of a gene having a 50 chance of being passed on, gene drives makes it close to 100%. Yeah.
Yeah, they're being tested in also mosquitoes to help eradicate malaria by spreading infertility or resistance genes through entire population. but they raise ethical and ecological questions. So deployment is tightly controlled, as it should be.
Yeah, and another big area is something called xenotransplantation, which basically just means transplanting animal organs to humans. And scientists are using CRISPR to knock out basically pig genes that trigger immune rejection, and the first pig-to-human heart transplant happened back in 2022. And as far as I can remember, it was a successful transplant at that. The individual is still living with that pig heart now.
Yeah, and by removing these immune triggers or adding human compatible markers, we get closer to solving the organ donor shortage, for example.
Yeah.
Which is pretty.
Yeah. So CRISPR also powers biomanufacturing, like editing yeast, bacteria, or mammalian cells to produce insulin, antibodies, biofuels, and even sustainable materials like lab-grown leather or spider silk.
Yeah. So some research models is that CRISPR has also been a game changer in basic science. So scientists can now generate genetically modified organisms much faster, like mice, zebrafish, or fruit flies, with custom edits that lets us study specific genes in real time.
Yeah, these models help us understand diseases like Alzheimer's, diabetes, or cancer, and also test drugs before moving to human trials.
Yeah, exactly. But what's remarkable is the speed and precision we now have, right? Before CRISPR, generating a knockout mouse could take more than a year. Now it's a matter of weeks.
Mm-hmm. So whether it's editing humans, plants, animals, or microbes, CRISPR is proving to be more than just a lab tool. It's a platform for redesigning biology itself. Now, we've gone over what CRISPR does at a high level, so in the more surface area in part 1. But now let's dig a little bit deeper into how it works on a more molecular level. We will try to keep it as clear and digestible as possible, but it's still a very complex topic.
Yeah.
So, CRISPR's most iconic move is using the Cas9 protein to create a double-strand break, or DSB, in the DNA. That means cutting both strands of the DNA helix in a very specific location.
Yeah, so picture DNA as a zipper and Cas9 as tiny scissor. So a DSB is like snipping through both sides of the zipper at once. It completely opens the code at that point. So now the cell panics and has to repair it.
Yeah, the cell has two main ways to repair that break. The first one is non-homologous end joining, in short, NHEJ. This is a quick and dirty kind of a repair method that glues the ends back together, but it's prone to errors.
Yeah, and those errors, like losing or adding a few DNA letters, can be helpful if your goal is to knock out a gene entirely, since the disruption makes the gene unreadable.
Yeah, so it's like fixing a cart jump rope by just knotting the ends back together without taking the alignment. So it works, but the rope doesn't swing the same way anymore.
Yeah, and that's when the second method comes in. So that's homology directed repair, in short, HDR. So if you give the cell a piece of DNA with matching ends called homology arms, the cell can use that as a template to repair the break accurately.
And that's what makes HDR useful for precise edits, like correcting mutations. The challenge is HDR mostly happens during cell division, so it's inefficient in cells that aren't actively dividing, like neurons or muscle cells.
So think of it like patching a ripped page. HDR is using perfect photocopy to restore the missing text, but the copier only works at certain times and not in every room.
Yeah. And one risk with CRISPR is that Cas9 can sometimes cut in the wrong place. So that's called an off-target effect. And that happens if other sequences in the genome look too similar to the target.
Yeah. And to minimize that, Researchers design guide RNAs very carefully and use computational tools to predict potential off-target sites.
Yeah, there are also modified version of Cas9 called high-fidelity Cas9 variants that are engineered to be much more selective. High fidelity here means Cas9 won't bind unless the DNA match is almost perfect, like a lock that rejects every slightly wrong keys.
Yeah, and the goal is to increase specificity. So you're cutting only where you mean to and not damaging unrelated parts of the genome.
Yeah, and it's the difference between a general pair of scissors and a surgical scalpel with fingerprint lock protection.
Yeah. Next, we have more advanced versions of CRISPR that don't cut both DNA strands. So one is called base editing.
Yeah, and base editing uses a modified Cas9 that only nicks one strand and fuses it with an enzyme that chemically changes one DNA base into another. For example, cytosine, as with the letter C, can be turned into thymine, or letter T.
Yeah.
So instead of cutting the rope and tying in a new piece, you are using a molecular sharpie to just change one letter on the rope. It's more precise and less disruptive.
Yeah, and then there is prime editing, which is even more versatile. So it also uses Cas9 nickase, a version of Cas9 that only cuts one strand, but this time it carries a built-in reverse transcriptase, which I will tell you what it is.
Yeah. So reverse transcriptase is an enzyme originally found in viruses. It's what HIV uses to convert RNA into DNA. In prime editing, it acts like a typewriter, writing the desired DNA edit directly into the nicked strand, and it's based on instructions encoded in the guide RNA.
So it's like a smart pen that knows exactly which words to fix and rewrites them on the page without needing to rip or replace anything.
The advantage, well, you can make insertions, deletions, and point mutations all without causing a full double-strand break, which reduces the risks of random errors.
Yeah, and CRISPR isn't just one tool, it's a growing family of tools. Scientists have discovered Cas proteins with different microbes, like Cas12 and Cas13, or even Cas theta.
Yeah, each of these has slightly different properties. So some are smaller, which is important because they fit better into delivery vectors like AAV viruses, but others recognize different PAM sequences. So those are the short bits of DNA next to a target that Cas needs to bind.
Yeah, so it's like building a set of screwdrivers with different heads, some tiny, some magnetic, some with longer arms. So you can open up any device, no matter the shape of the screws.
Yep, and then there's the delivery method. Like, how do you actually get the CRISPR into the right cells?
Yeah, so one breakthrough was the use of lipid nanoparticles. So like I said before, those are tiny fat bubbles that can carry CRISPR components safely into the cells. And they are especially good for delivering to the liver, and newer versions are targeting other tissues by adding molecules on the surface that act like zip codes.
Yeah, another method is transient delivery, where CRISPR is introduced as a ready-made protein plus RNA complex called a RNP. Since it doesn't hang around long, the risk of off-target effects goes really down.
Yeah, and CRISPR can also edit multiple genes simultaneously, a process called multiplexing. And that's important for studying complex traits that involve multiple genes, like cancer resistance or engineering crops to be heat and pest resistance at the same time.
Yeah, like imagine editing not just one word in a sentence, but a whole paragraph, changing punctuation, grammar, and phrasing all in one go.
Mm-hmm. Yeah, so, but why all these tools matter? Well, all these innovations, like high fidelity enzymes, base editors, nanoparticles, whatever, have a shared goal. Better precision, higher efficiency, lower risk, and also wider applicability.
And that's what makes gene editing increasingly realistic, not just for experiments, but for treating real conditions in living people.
Yeah, the next big step isn't just asking how we can edit, but when and why, and who gets to decide. In other words, the ethics of it.
Yeah, and that's exactly what we'll go into next. So, editing tools like CRISPR, base editors, and prime editors are extremely powerful. And that raises deep ethical considerations about when, where, and how to use them.
Yeah, and one big issue is equity and access. So right now, advanced therapies like hexagamglogene autotemcel, which edits the HBV gene to treat, the sickle cell disease and beta thalassemia, are life-changing, but they're incredibly expensive and also often only available in a handful of clinics worldwide. So who benefits? Wealthy patients in wealthy countries? Or are we just building something that everyone can access? Yeah.
It's like inventing a fire extinguisher, but only selling it in penthouses. The people who need it most, who often face the worst health outcomes, cannot get to it. And that applies to agriculture too. CRISPR edited crops that resist pests or drought could help small farmers, but only if the technology isn't locked behind expensive patents.
Yeah, and this ties into the larger question of intellectual property. Some biotech companies tightly control editing tools through patents and licensing, which can slow down research or block low-cost treatments. There's this ongoing debate: should gene editing tech be treated like open science or like a commodity?
And then there's the big red line for a lot of people, germline editing. So editing embryos or reproductive cells means the changes are passed to future generations who cannot consent. And that's a much heavier ethical weight than treating a disease in an adult. So even if we could do it safely, should we?
Some argue it's our moral duty to prevent severe genetic diseases. Others see it as playing God. But it's not just about the technology, it's about values. What counts as a disease? Is deafness something to fix or part of someone's identity? Different cultures and communities answer these questions very, very differently.
And beyond humans, we also have to ask, what happens when we release edited organisms into the environment? Whether it's a CRISPR mosquito to fight malaria or a heat-resistant crop, ecosystems are complex and very delicate. Some changes can ripple in unexpected ways.
Yeah, and there's a risk of unintended effects on pollinators, on soil microbes, and also wild relatives. And the people affected by those changes, like rural communities or indigenous groups, are often left out of the decision making. And it's not just about biology, it's also about justice and inclusion.
Yeah, another layer is biosecurity. So CRISPR isn't hard to access anymore. If someone wanted to misuse it, say engineer a dangerous virus, the tools are already out there. So there is a growing push for stronger oversight and researcher training on dual use risks.
Yeah, and this field also makes us ask what it means to be human. Are we headed towards a world where we start editing not just to treat diseases, but to enhance traits, better memory, taller height, muscle growth, higher intellects? Where's the line between therapy and enhancement? Who gets to draw it?
And then there's also a question, who gets left behind? So we risk creating a new kind of inequality, not just economic, but also biological. So imagine a world where some people are edited and others aren't. That's not science fiction anymore, it's an ethics problem.
Mm-hmm.
So yeah, gene editing is powerful, but it's not just a scientific question. It's a social and moral conversation that has to include everyone, not just scientists and policymakers.
Exactly, and we have the tools to change DNA, but that means we're also shaping the future of humanity. The real question is, how do we make sure that's a future worth living in for everyone? And that's not something that we can answer, us here in this podcast.
Yeah, and it's definitely something that you need to answer it for yourself.
Yeah. So we're getting close to the end, so let's start by recapping the big picture. So
Yep.
gene editing roots trace back to early genetics and recombinant DNA technologies back in the 1970s, which paved the way for transgenic organisms and the first gene therapies in the 1990s.
Exactly, and it's important to distinguish gene modification, which involves broader, sometimes less precise changes like inserting foreign genes. So, from gene editing, where we make targeted, precise changes at specific DNA sites.
So before CRISPR, we had tools like zinc finger nucleases, ZFNs, and TALENs, which showed it was possible to cut DNA at targeted locations, but these tools were complicated and costly to design and implement.
Then,
Yeah.
CRISPR-Cas systems revolutionized the field by using a simple RNA guide for targeting, making editing faster, easier, and allowing multiple edits at once. And the technology keeps evolving with Cas12, Cas13, and newer bases and prime editors expanding what's possible.
Yeah, there is also a big challenge as well. So, depending on whether you're working ex vivo or in vivo, options range from viral vectors to lipid nanoparticles to directly delivering the ribonucleoproteins.
So applications are incredibly broad, from treating blood disorders and eye diseases to cancer immunotherapy to improving crops and livestock to developing research models and diagnosis. Some landmark milestones include the FDA's approval of Cascavy in December 2023 and the first pig to human organ transplant using gene edited organs.
Mm-hmm. But alongside all the excitement, there are crucial ethical, regulatory, and social questions. Germline editing remains off-limit worldwide for now, while somatic therapies proceed carefully under oversight. We need ongoing attention to equity, environmental impacts, biosecurity, and public trust.
Yeah, I would say looking ahead, we can expect more precise editors, better delivery technologies, and also broader applications. But success will depend just as much on building robust ethical frameworks and also ensuring the equitable access.
So yeah, I definitely agree, and it's an exciting and complex frontier, and staying informed and thoughtful about both the science and its implications will be the key as this field moves forward.
Yeah.
Yeah.
So that wraps up our deep dive into gene editing and modification. We've traced the history from early recombinant DNA to the CRISPR revolution, seen how the tools work, and explored real applications and future directions, and discussed the ethical landscape.
Yeah. Thank you for listening to Breakdown of Science. I hope you guys enjoyed this episode. If so, please follow, comment, and share.
And also, as we approach the end of our series, of our podcast, this is actually our second to last episode. We'd love to hear your thoughts on gene editing. How do you feel about CRISPR-based therapies in medicine? If you had a serious genetic condition, would you consider such a treatment?
And what about gene editing in agriculture? Like, should precise genome edits be viewed differently from older GMO approaches? So we are really interested in your perspective on that.
Yeah, plus a big question, where do you think society should draw a line on human enhancements? What kind of governance models would be fair and effective, especially on a global scale?
And if you have any questions or thoughts, feel free to comment or send them to us on our Instagram. We'll be doing another Q&A episode next week, the final one in the series. So make sure to follow us on Spotify, YouTube, and Instagram to get notified when it drops. So see you next week. And thanks for listening. Bye.
Yeah. Bye.
Bye.